It is October 15th. We are here in week 8 of the semester. All right. So, yeah, we're halfway through. And once we get into November, we got a couple vacations coming. So let's just keep on making it through the semester, you know? Week 8, no quizzes, just read stuff, The Secret Life of Everyday Things, and continue collecting citations for your annotated bibliography. Don't forget about that assignment. You don't want it to sneak up on you and then be stuck doing it on like December 5th. It's too much work to do in a single weekend, but you do 3 citations, 4 citations each week. That's manageable, you know. Anyway, that's just a quick check-in. The next quiz will cover the material that we're talking about in week 7 and week 8, the week 7-8 PowerPoint. It's hidden in the week 10 module. That says November 12th, but I'm almost positive it's actually November 2nd. That's a classic typo. Let's fix that right now because I don't want to mess everybody up. The official due date and the schedule should all say November 2nd, but my apologies for that if that's caused anybody confusion. November 2nd is correct. Okay, just fix that real quick. Cool. And confirm that the official due date is actually correct until November 2nd. Perfect. Also, don't forget about weekly discussions. Participate in those weekly discussions, get your 3 points. Any questions before we go ahead and dive back into our biodiversity PowerPoint? Awesome, let's just get into it. So, we've been talking about conservation. I'm going to adjust the lights. Brace your eyes. We're going back lights. Turn off the front lights so the screen looks a little better. I like that much more. So if you recall, we've been talking about a lot about diversity, all right, about biodiversity. Since the beginning of the semester, I've been trying to emphasize like the word diversity means something very specific to ecologists. And we need to understand how environmental scientists and ecologists measure diversity. This lecture right here has been, my intention has been to delve a little deeper into some other aspects of biodiversity and then how we conserve biodiversity. And that's where we sort of are right now. The remainder of this is mainly talking about different types of conservation projects and habitat conservation. So we wrapped up talking about some, species to prioritize for conservation, introducing the concepts of the umbrella and the flagship species. We've already spoken about keystone species and how they're good targets for conservation because they're essential for healthy ecosystem function, for healthy community interactions, right? Now, other targets for conservation, the umbrella and flagship species, are a little more pragmatic, where flagship species, there's usually a lot of public support for conservation efforts because they are charismatic, they tug at people's heartstrings, you can look at them and say, yeah, I don't want that to go extinct. There's a giant centipede on this chain of islands off of Australia that's endangered, and it's a little harder to get people really excited about a centipede this big when people are holding it like, see, it's cute, It's a little more difficult. And I don't blame them, all right? No. So we've got the concept of the flagship species and the umbrella species as good targets for conservation efforts. Usually, if you can get species-based conservation efforts implemented, you end up protecting more than just that species. And nothing is more exemplary of this than nothing's a better example of this than the umbrella species, okay? Umbrella species are identified as, if you protect them, okay, you're protecting a lot of habitat and therefore a lot of other species. So it includes species that have a very broad range or species that, say, consume a lot of different prey. You have to conserve the prey population for the predator population to be protected. How do you conserve the prey population? You conserve the whole habitat. Blah, blah, blah. You get the idea of the umbrella species here. It's all right. All right, cool. Recovery plans, these are what you've actually published that say, hey, here's what we're going to do to help the species recover from the endangered status. And it includes all of the different ex situ or in situ conservation efforts that you might be putting into place. And these plans do need to take into account economic impacts, all right? If there's a commercial effort that's going to have a societal benefit, whether it's economic or quality of life wise, that's going to be weighed against conservation of or preservation of or maintenance of the population of the endangered species. There's just no two ways about it. All right? So compromise may be necessary. Private land makes it more complicated. I talked to you a little bit about this. We should have some more examples of this when we hit our ecological economics PowerPoint, because I'm going to talk about how private land works and how private land owners can, say, set aside some land for conservation efforts in order to continue doing non-sustainable practices or things that are going to impact an endangered species. We'll talk about that in ecological economics a little bit. But now we're into new material, and we are talking ex situ conservation. So are we all comfortable with the difference between in situ and ex situ conservation projects? Refresher. Awesome. Thank you. It was just a trick to get some engagement from the class at 9 a.m. I was like, are you comfortable with this concept that I know we're not comfortable with? Yeah. Good. Good deal. Thank you so much. In situ, it means in sight, at the location, in the natural location, right? Ex situ means out of the site, out of the natural location. So an in situ conservation project or an in situ, a recovery plan that includes in situ efforts, it could have things like, we are going to issue hunting permits for deer species because the deer are consuming this endangered plant species too much, and that's going to drive that endangered plant species towards extirpation, which is localized extinction. That's in sight. It doesn't require you to move anything out of its natural habitat. Ex situ conservation projects specifically involve you removing individuals from that site and then fostering that population's growth or fostering breeding efforts, fostering the biodiversity at another location. Okay? So establishing a nature preserve, for example. We did our preservation versus conservation talk a little bit ago. Establishing a nature preserve is always an in situ conservation effort, right? Because by necessity, it happens in the natural site. Ex situ could be like you take some animals and you put them in a zoo that has a captive breeding program. That's ex situ. Or you take some animals from one island where they are, let's say there's a virus spreading through a population on an island. These are real-world examples without species names thrown on them to try to keep it simpler. Right? You got an island population, and there's a virus spreading through that population. There's an adjacent island where that population does not have the virus problem. Maybe the population's not at a high enough density. So you remove individuals that are not infected to that other island, and that increases genetic diversity. And then after you let the island's population sort of perish, the virus runs its course, you move those individuals from the island, from that other population, back to the island, all right? So that's an ex situ conservation project where you're removing individuals from their natural site, their site of origin. Solid? In situ, ex situ. So we'll talk about some ex situ conservation projects now. Bullet points, right? They're removed from their habitats, established in breeding populations, or you could be moving individuals into like a gene bank All right, like you can take gametes and move them. You can collect sperm cells from animals or egg cells from animals and then store them somewhere. And so you're banking genetic diversity. Okay. At the end of the day, the goal of the XC2 conservation project is supposed to be reintroduction, rewilding, right? You put the animals back out. We'll use our condor example as an example of a very effective XC2 conservation project. All right. Now, this last bullet point here, not every breeding or captive population was necessarily established for XC2 conservation purposes. There have been a few species that were thought to have gone extinct and then were found in essentially just rich people's private zoos. Like someone's really into beetles, and they're like, I got these cool beetles. I had them imported from some other country, and yeah, it was illegal, but that was a while ago. And now I got the only beetles of these species. People be like, oh, that's cool. Can we breed them and reintroduce them to the wild? And sometimes, yeah. Anyways, so this last bullet point is just saying, not every XC2, not every breeding population was necessarily established for conservation purposes. There's some fun examples. Check it out. Look into it. It's really cool. Okay? Cool. So, more details of different types of conservation, of XC2 conservation projects. We've got bespoke and intensive captive breeding programs. All right, so these are designed for an individual species. It'll be an effort to say, let's go with the condor, right? You have a California Condor Conservation Project, and that California Condor Conservation Project is designed to capture condors, breed condors, and then eventually re-release the condors. It's a condor project. It's bespoke to them. You've also got general breeding programs, and the example of this is a zoo. The zoo has a bunch of different animals. The aquarium has a bunch of different animals, all right? Botanical gardens have a bunch of different plants, and they will usually be carrying out breeding efforts, either through exchanges of animals between zoos, between aquariums, between botanical gardens, exchanging seeds, all right? So they're carrying out some sort of general breeding program that's more widespread across the species that they're fostering, let's say. Okay. Cool. Then we've got gene banks, and gene banks don't really fit this first bullet point. Just one sec. This first bullet point here of establishing those breeding populations, because a gene bank is not a breeding population. It's cold storage of gametes or genetic material. Seed banks are gene banks. You got egg banks, sperm banks. All these things are gene banks where genetic material is being kept safe so that it can then be reintroduced into a gene pool later. Yeah, question, please.
So general breeding programs aren't trying to conserve ecosystems, right? They're just species.
I see, yeah. So usually, here's the difference, right? The intensive captive breeding programs and the bespoke programs, their only goal is we have one species and we are going to focus all of our expertise and efforts on breeding it and making sure it has diversity and then getting it back into the population.
So trying to make it so it could survive in the wild on its own.
Yeah, that's the goal, yeah. But a general breeding program is something that it's usually another aspect of a zoo or an aquarium or something, or a botanical garden, where they have a general breeding program where they routinely breed the animals or the plants that they have there. It's not as specialized.
And they're not so it's not trying to go to a specific goal. They're just kind of breeding them.
Yeah. Now there may be a specific goal, hey, we know that this species is endangered in the wild, so we breed them to make sure that we've still got a population. But also it might just be a breeding project to keep animals in the zoo. So you don't go out and capture new animals. That's not as much of a it's still conserving the genetic diversity because you're keeping more individuals alive, and a general breeding program should prioritize limiting inbreeding. That's why you do the exchanges of animals between the different zoos, right? But it's not as intensive, and it's not the only focus. So there's a note here on the bullet point even about how they're balancing economic interests along with the breeding interest. You want to have a good zoo, and then you justify that by saying, oh, that increases public interest in nature. It raises awareness in the public of these species, and that's going to make people more likely to donate money or time to conservation efforts. It's the classic line that you respond to anybody who's like, a zoo's an animal prison. That's the classic rebuttal. Is you go like, well, no, it increases, public engagement with animals and with nature, and some people would never see this animal or know that it exists without the zoo. Is that a bad argument? I don't necessarily think so. It's just a very rote argument that you hear a lot. All right. So, did that answer your question all right?
Yeah.
Solid. Okay, cool. So, hey, check this out. Here's this Amur leopard. That's a cool looking cat right there. All right. There's only 30 to 35 individuals estimated in the wild. All right, but there's 180 in captive breeding populations in different zoos. So there's more in captive breeding populations than there are actually out there in the wild. And with 30 individuals, is that enough genetic diversity for a sustainable, stable population in the wild? What do their habitats look like? If they're that small of a population, you can rest assured their habitats have become fragmented and shrunk and are under threat. And there's probably some taking going on where people are hunting them, It's definitely some stuff going on in the wild that might make that wild population unsustainable, but it's possible with very active management of those 180 captive individuals, you might be able to maintain enough genetic diversity, increase survival rates of offspring enough to reestablish the population. All right? Good. So this shows sort of the overlap, okay, between a general and an intensive captive breeding program where there are all of these zoos that have these leopards in them, and they're involved in sort of a group intensive captive breeding program, right? Even though each zoo has other interests that it's balancing and it has its own general breeding program, the zoos also collaborate to try to maintain diversity in this population. Okay. So, why do you do ex situ conservation projects? Well, you might just be trying to develop an insurance population. You've got a species that's endangered or at risk, and it exists naturally in a habitat that's at risk. You know that there's activities going on, or like the animal is regular, there's poaching of the animal, or the habitat is being destroyed really aggressively right now. So you take some animals away, and you establish a little insurance population. Or as you'll see in the example of the Tasmanian devil, there's a virus that kills the Tasmanian devils like crazy. It's real bad. So what they'll do is they'll take uninfected Tasmanian devils and put them into an ex situ breeding program so that if all of them got wiped out by a really bad virus, they could be reintroduced. It's an insurance population. Sometimes you are affecting a temporary rescue, which would be, hey, there's a fire. There's a big fire coming. We know, we have historical patterns that say it, and we have models that predict that in this year or the next year, there's going to be a massive fire that sweeps through this area. We're going to just effect a temporary rescue and remove those species for a while and then reintroduce them once that disturbance has passed. Okay? Cool. We talked about, sorry, I'm not laughing about the animals dying. I'm laughing because of the loud drilling noise, which, it makes so much sense to do that work at 9 a.m. on a Wednesday, It's the only time they can do it is the thing. It's the only time. Anyway, a lot of these have overlap. A lot of these ex situ goals are pretty intuitive, right? The population's at risk in its natural habitat, so you move it out. If the population goes extinct in the wild, that's one of those categories of extinction that we've spoken about, you might have enough individuals in captivity to keep that species alive on the planet. So you're still maintaining biodiversity globally by having one more species on the planet, okay? Good stuff. Manipulating demography, okay? So this, and you'll see many, when we think about conservation, I'm hoping that I've been sort of laying this out for you in a way that naturally progresses. When we think about conservation, it's really standard for us to think about preservation, all right? That, all conservation efforts involve is don't harm that animal, let it just do its thing. But that's preservation, okay? That's a nature preserve, not a conservation effort, okay? Conservation can be really, really active. Sometimes it involves taking animals out of a population. So this is a very interesting example here of once animals have passed reproductive age, they're no longer contributing to the growth of that population, right? They're no longer contributing to maintaining the genetic diversity of that population. But I'll tell you what they are doing, and that is consuming resources that the reproductive individuals could possibly consume. So intra-specific competition limits population sizes. And if we can reduce intra-specific population, we can let the population start to grow a little more rapidly. And one way to do that is by removing individuals post-reproductive ages. So this is one of the justifications for allowing trophy hunting of specific individuals. Say like, well, that individual, you can go out and you can hunt that lion because it's not reproductive anymore. You go out there, it's got a tag on it. We'll have a guide with you. They'll point out the lion, and then you shoot it, and you give us $50,000, and then we put that back into lion conservation efforts, and we've reduced one of the intra-specific competitors, and hopefully the population grows again. All right. So when I say removing individuals at certain life stages to reduce mortality, that's the best example of that bullet point there. And that's a real thing that is done. All right. It's pretty wild stuff. Oh, but I'm sorry. I got a little bit sidetracked because our title on this slide is XC2 conservation projects. Same thing would apply if you took the post-reproductive individuals and move them to a zoo. All right. It would be in C2 if you issue a hunting permit to kill a certain lion. It's XC2 if you go and get that lion and put it into a zoo. See what I mean? So, my apologies. Just got a little off a little tangent there. All right, great. Reintroduction's usually the goal of these XC2 conservation projects. Assisted colonization is something that we've kind of touched on, where we talked about translocating plants when we talked about this, I believe, right? How plant movements can be slower than climate change, where if the plant was 100 miles north, it would be doing great. But it's instead experiencing drought and heat stress. And the plant population can't move that rapidly because plants don't walk, okay? But assisted colonization, okay, would be an example where you take some animals or you take some plants and you just put them in a temporary XC2 population, and then you translocate them. You're assisting them in colonizing a new area. This involves a lot of concepts like climate envelope modeling or trying to figure out which community types are going to be adapted best to changing conditions, because conditions are changing more rapidly now than in the past, and therefore natural migration rates, natural movement rates of community may not necessarily keep up with the rate of changing conditions. So translocations and assisted colonizations can be really useful. Also, hey, you can do research in an XC2 population. Cool. Great. Love it. Education and awareness, yeah, that's how you justify every zoo, you know. Okay, so check it out. I'm bringing back the IUCN again. So everybody references the IUCN. I already mentioned to you that, maybe it's gotten a little bit shady in some of its dealing. That's really big and maybe possibly a little bit untenable to manage all those moving parts. But everybody still references IUCN guidelines. This is from a different textbook. This is from an applied ecology textbook where they just say, okay, hey, conservation, here's the guidelines for conservation that everybody on the globe follows. And they just leave it at this. And then they're like, okay, all right, well, cool. These are the guidelines. So this is a little screenshot of the IUCN's XC2 conservation guidelines. All right, when is it okay to take the population out? And it's always going to require some sort of survey, all right, that weighs the cost of the effort with how likely it's going to be how likely success is of the effort, okay? There's also the impact of removing individuals from a population, which needs to be balanced. So these are pretty reasonable, good guidelines, right, that they've laid out. You got to figure out what's the potential net positive, how likely is it that the potential will be realized, and then also, what's the negative impact? And then they throw this in here. This will be a wise application of the available resources, which is, an economic question. If we have only so many resources, and is this project likely to succeed? What's the best benefit? What's the best possible outcome if it does succeed? Pretty common sense stuff. All right, let's talk about the California condor example. So, 1987, they go out, they capture every wild condor. We're assuming, records indicate that they probably got them all. It's a big bird. You know the condor? It's like a big vulture. You've seen it. It's massive. There's some reasons that it's not super adapted and that other birds do better than it. It doesn't have the same sense of smell that other species of vultures have. Well, it's not a vulture, but that other species of, oh my gosh, I'm blanking so hard. Some animals eat other dead animals. What are they called? Decomposer. I'm looking for a specific one. Yeah.
I know.
Huh? A scavenger. Scavenger, yeah. Scavenger's good. There's a word for when animals feed only on carcasses. Yeah, a carcivore. I don't know. I'm forgetting the word. It's so embarrassing. No. Yeah. I know. In week 3, I brought it up even. I was like, oh, here it is. It's this type of feeding habit. We'll figure it out later. Or we'll just keep moving forward and eventually we'll forget about it. Exactly. As you're all walking out the door, I'm like, don't go. Nah. I thought I could just sneak up on it. Okay. All right. Hey, listen. There's some birds that eat dead animals. They eat carcasses. There's a name for them. Sometimes these animals use smell to detect the carcasses. And those animals didn't need a dang recovery plan to be successful in the wild. Animals that rely on sight to locate carcasses, like the condor, they have a bit of a disadvantage. All right? So they're kind of naturally outcompeted by other species of herbivores. All right? Cool. But conservation effort was implemented, a recovery plan was implemented involving XC2 management. All the condors get captured. They all get taken into an intensive breeding program, a bespoke breeding program that's only for condors, where condor experts reside, and all of the funding for that facility goes towards condor conservation. All right? Makes it a bespoke, intensive program. They put them in some zoos. So it's an intensive breeding program going on at zoos. And then what they did, which was really fascinating, is they took advantage of a natural aspect of the condor's biology, which is that they can double clutch. And what that means is they can lay a set of eggs, all right, and then if they don't have, if the offspring perish, they will lay another set of eggs, all right, in a single season. So theoretically, reproductive output of the condors could be doubled. All you have to do is let the baby be born and then go over and just take it and then not kill it, keep it alive and raise it to be healthy, and then they'll lay more eggs. So this aspect of the biology, you know, it was utilized to ramp up reproductive rates. You got this XC2 population, and it was very, very successful. Condors got reintroduced. The condor population's genetic diversity seems to be viable. Population viability analyses look positive. They did a lot of specific things, like they fed the condors with the babies with these condor puppets to try to limit human habituation, it's called. You don't want the animals that you're going to reintroduce into the wild to become used to eating from people or to get too comfortable around people, because I don't know. I mean, there's a big bird. It swoops down on you because it thinks you're going to feed it something. Maybe you whack it with a golf club or something. And that's no good, all right? So the reason that we talk about this specific program is take a look here. This program's success relied on a thorough understanding of the condor's life history and its biology and all the specifics here. So it took a lot of collaboration with experts and then implementing a recovery plan, which wasn't just as simple as just put like 15 of them in a big aviary and then like wait for the babies to breed and then put the babies out into the wild. You see, it required an understanding of the biology. Anyways, I just love the puppet right there, the condor puppet. It's very cute. The condor puppets for this project, that's my understanding from reading about this project. But feeding babies in XC2 conservation projects with puppets or without direct human interaction is a classic, you know? Or at the very least, you shove the food through a door and you don't actually like see the animal, even though, you know, you'd imagine anybody in that job probably does want to pick up the animal and pet the animal. So maybe this is more for the researchers and the conservationists so they still get to interact with the baby condor. Because who wouldn't want to interact with that thing? Look at it. All right. Here's another cool aspect of how these projects, the concept all sounds so simple, but there's things to keep in mind if you're trying to implement these recovery plans. Like, remember those IUCN guidelines? You need to have some idea of how likely it is that the project will be successful. And I'm going to hit that point again and again. You got to have some way to demonstrate that all this money, all this power that we're going to be using to carry out this conservation project, and the risk that we take by removing individuals from an endangered species out of their natural habitat, that all of that has any likelihood of success. So what they did is they took these Andean condors from South America. It was a captive population, all right? They released them and they recaptured them. They were captive bred, all right, with the same sort of procedure, but the species wasn't endangered. So you could do this before you started re-releasing the captive bred endangered condors, okay? And you had an idea of, oh, what do our success rates look like? What are the survival rates like? Was the population gonna grow? Is it gonna decline to nothing? So you did some sort of pre-release, right? But of a different species that was very similar to see if the efforts would work, all right? It's just more ways of demonstrating likelihood of success. which is one of the guidelines for these conservation projects. And it's not just lip service to say, oh yeah, I talked to an expert and they said it would probably work. It's like, no, there needs to be some data. There needs to be something to demonstrate possible success. Anyways, it only cost $35 million, but now the wild population of California condors is over 400 individuals. That's a pretty good deal. It's like $100,000 per bird. Not so bad, right? So if you capture a California condor, you get 100 grand. That's the policy, dead or alive. So get out there, students, and happy hunting. Not true. People seem to like the gene banking topic. People seem to like the seed banks. These facilities are really cool because they gotta be cold. And if you know anything about energy and energy use, a lot more energy is spent keeping things cool in our modern world than keeping them warm. All right? We'll talk about that when we get into later lectures about energy. We'll talk a little bit about like cities and how much is spent cooling buildings versus heating buildings and natural systems that could reduce cooling costs. But keeping things cold is hard, all right? And keeping things cold helps to prevent them from decaying, okay? So, gene banks, all right? Gene banks are just stored, as you say right here, stored biological material. So it can be embryos, okay? It can be seeds, all right? It could be sperm cells, all right? Plants are usually easier to manage in this way because plants develop seeds as an adaptation to make a little thing that can remain viable until conditions are right. The whole reason to have a seed with stored resources and dormancy and all these concepts is so that you can release the seed and then it can wait a little while before it actually germinates. A lot of important things there. Some seeds have different dormancy mechanisms. It's all really important stuff, okay? So this means that sometimes you just take a seed, you keep it dry, you put it in the fridge, it could last for, 100, 200 years, and you've successfully banked some genes from that population. So that's pretty nice. Here's an example of a seed vault, the Svalbard Global Seed Vault. It houses seeds that are significant for food production. That's its primary focus, which is pretty cool. The permafrost there, and its location, mainland Norway, North Pole, around there. It's up north, right? There's permafrost. It's always frozen. It's real cold there. So it's a naturally chilled seed bank, all right? And they've got the focus of agriculturally important food crops. Because, what was that movie where Matthew McConaughey's in a watch or something? Interstellar, right? He gets trapped in the watch and he's like, "Let me out, my daughter. I'm in the future. You have to send me to space." Remember? Yeah. He's in a bookshelf? What happens in Interstellar? Didn't it win an Oscar? Everything's so difficult. The point of the matter is Matthew McConaughey lives in the Svalbard Global Seed Bank in Norway.
It's okay, nobody watched the movies.
Good. That's totally fine. The thing is pretty forgettable, quite frankly, if my experience is telling you anything. Why did I bring that up?
Or are you mixing Interstellar and Inception?
No, but those are both Christopher Nolan movies, right? So that it all comes together. Wow. This picture? It's possible.
I don't know. Maybe I did watch that movie.
Oh, you know why? I remember, he doesn't live here in the movie. What happened was all the crops died because there's some virus. And the only thing they can grow now is potatoes or something like that. And at the beginning of the movie, he's like, well, the corn virus just killed all the corn. Now all we have is potatoes, so I'm going to go to space. And that's the movie, all right? But if there's a virus that sweeps through and kills all of the modern corn, well, thankfully, there's the Svalbard Seed Bank, where old corn genetics are being conserved. I tied that up perfectly. That went great. That was perfect. I'm so happy. It's all recorded. Someday they can play this at my hearing. All right. Hey, check this out. The Kew Gardens. You ever see these Kew Gardens? They're pretty cool. All right. They've got a seed bank as well. The Millennium Seed Bank Partnership, coordinated by Kew Gardens in the UK. Look how cold this person is. Oh, brr. What their goal is to collect 25% seeds from 25% of all known flora. They're just banking seed, banking genetics here, right? But this is not a naturally cooled space. So every once in a while, with seed banks like this, there'll be an issue with energy delivery, and they'll lose a ton of materials. All right? So, pros and cons. Yes?
Can't a lot go wrong here?
Oh, yeah.
If there's a power outage, like, one of them can just die.
Yeah, absolutely. There was one, I wish I could remember the name of it, but there was one seed bank where that happened, and they basically lost like half of the seeds that they'd banked, and it was a whole thing. But then some of these things will remain viable even after the power outage, and there's redundancies built into these systems to protect against those sorts of situations. But yeah, if you're centering all of your material in a single location, there's a lot that could go wrong. Absolutely. But yeah, it's pretty interesting stuff. Every once in a while, they do viability analyses in these places where they take a seed or 2 seeds out of the collection or 10 seeds out of the collection, and they sow them and see what proportion of them actually germinate, and that's how they make sure that the seeds remain viable. And if they're no longer viable, they go and collect more of that species. And that gives them a sort of an idea of how regularly that species needs to be collected. And now you have a historical record, and it all just keeps moving forward. So this MSBP site, that's the Millennium Seed Bank Partnership site. They're stored at -20, all right, in these glass containers at a specific humidity. Special samples that are high priority get stored at an even lower temperature in liquid nitrogen, and they do these germination tests that I was mentioning previously. So that's how they sort of maintain those seed banks like that. Animal gene banking is trickier because animals don't make seeds. Some animals make eggs, but it's not the same thing. So usually what gets banked are sperm cells, spermatozoa, okay? One of the benefits of this is that you can thaw those sperm cells and use them in fertilization of a naturally occurring population. So you thaw out the sperm 20 years later, and then you inseminate a wild population of individuals, and now you've just taken genetics from the past and brought them into the present. And so that's going to be more, there could be some genes that were lost. There's going to be a lot of genetic diversity and differences between those. Now, the viability of sperm cells is not usually great over very, very, very long periods, but they're the most stable cells for this purpose. Yeah. So usually it's a big animal sperm bank, which is kind of interesting. All right, so there's banking. That's an ex-situ conservation effort. Translocations, let's talk about them. Translocations are any effort, a translocation is any effort that moves species from one area to another area. All right, so as I mentioned before, hey, you could move species away from a threat. Oh, I didn't include this ecological impact assessment. My bad, all right? So that reference should have been removed from this bullet point. That's all on me, absolutely. Anyways, translocation can be done for a number of different ways. A temporary rescue, assisted colonization, these different things that we've already sort of spoken about. But overall, the term is a translocation for a movement of individuals. Any questions about anything here? Cool. The Tasmanian devil example. Here we go. It is the largest extant marsupial carnivore. Extant just means that it exists, that it's alive currently. It's the opposite of extinct. So it's the largest extant marsupial carnivore. It looks like that, all right? A super charismatic creature. Look at it. It's really adorable, okay? They only exist on Tasmania naturally, hence the name, Tasmanian devil, all right? There was a small population that colonized the area, and so if you remember our genetics, well, not really our genetics talk, but our evolution talk, this is an example of an immediate, of a founder effect, okay, where a small population establishes there's a small founding colonizing population that establishes and then grows in size. So it already had a reduced gene pool. And then there were a bunch of bottlenecking events where the population shrunk, okay? This leads to reduced genetic diversity, inbreeding depression, heightened disease vulnerability. We've already talked a little bit about inbreeding and how it's pretty complicated, why it actually leads to negative characteristics. But if you think about a disease, right? If a disease can get by your defenses and you're very closely related to all the other individuals in your population, they can probably get by their natural defenses as well. Just that straightforward. More similar population, more likely to be susceptible to the same disease. Cool. So check this out. Devil facial tumor disease. It's a transmissible type of cancer. And if that doesn't freak you out, like I don't know what will. Transmissible cancers. It's terrifying. It's like learning about prions when mad cow disease was such a big thing, and then your whole idea of what a disease or a pathogen is shifts. But the idea of a transmissible cancer is absolutely terrifying. And it can transmit so rapidly through this population because their genetics are so similar. So this was a major issue. It looked like the Tasmanian devil might go extinct. They set up an insurance population, and they view this as an arc. It's not a very desirable preservation strategy to just set up a little population of a few individuals and be like, hey, we got some, all right? But it's the insurance population that's currently established. And we'll come back and talk a little bit about Tasmanian devils on Friday. And then we'll just talk more about conservation projects, landscape conservation. It's going to be a ton of fun. Thank you all for making it today. I hope you have a good rest of your Wednesday. And remember, folks, we are past the halfway point. So we just got to make it less time than we've already made it. And you already did this, so just do less of it. Again, that's it. That's it from me.